A mitochondrion contained in a 1/2 micrometer thick section of chicken brain is viewed with a high-voltage electron microscope. When the section is tilted in the microscope, it can be viewed from many different angles, and a large amount of three-dimensional detail becomes apparent. Images from such a series of tilted views can be used to calculate a three-dimensional reconstruction, or tomogram, of the mitochondrion. The tomogram of the same tissue slice is shown here as a series of stacked images. The movie steps through the images one by one, from the bottom of the stack to the top and back. This allows us to trace individual membranes in 3 dimensions. To create a 3-dimensional model, membranes in an individual slice of the tomogram are traced. In this case, the inner membrane is traced in light blue, where it parallels the outer membrane, and traced in yellow, where it folds into the cristae that protrude into the mitochondrial interior. The tracings from all sections are then modeled as 3-dimensional surfaces, and displayed as a 3-dimensional model by a computer program. Such a model can now be viewed from any angle. In this view, only 4 cristae are shown and the others are omitted. The cristae are colored differently and show the variety of shapes and connections to the inner membrane in a single mitochondrion. The model also shows the reconstitution of the outer mitochondrial membrane, represented in dark blue, as well as 2 fragments of endoplasmic reticulum. Regions of such close proximity between the 2 organelles are quite frequently seen in cells. Note that there is no continuity between the mitochondrial and endoplasmic reticulum membranes. Lipids are thought to be shuttled between the 2 organelles by special carrier proteins that operate in this gap. Dynamic properties of the mitochondrial network can be seen in this living yeast cell, which expresses the green fluorescent protein GFP fused to a mitochondrial signal sequence. Membrane fusion and fission events constantly reshape the organelle. Using a confocal microscope, an optical slice containing only the top focal plane of the cell is recorded in this movie. The remainder of the network is out of focus.